Alright guys, welcome back to Underground Science. In this video we're going to be talking about a nonsense mutation, okay? So if we see here we have a 3' to 5', right, DNA strand. So we have TAC, CTT, GTT. If you transcribe that, right, we get AUG, GAA, CAA, okay? If you translate that and go to your codon chart, you'll see that AUG codes for methionine, GAA codes for glutamine, or glutamate, and then CAA codes for glutamine. Okay, so once we know that, what we can do is, if you want to make a nonsense mutation, right, what if we went here and we changed this cytosine into adenine? Okay, so let's go and do that. Let's say we have a mutation, and that mutation leads to this cytosine changing to adenine. Okay, let's put an adenine here. So we have a cytosine to adenine mutation. Okay, so now let's see how that affects us. Okay, so if we transcribe this strand now, instead of this being GAA, right, it's going to be UAA. Okay, so let's go and erase this. Okay, we're going to have a UAA here now. Instead of this being a glutamate, right, UAA actually codes for a stop codon. This basically, in translation, UAA, we'll see later when we cover translation that a release factor comes and binds here when, obviously we'll have ribosomes and everything, right? But the release factor is responsible for binding to the stop codon, okay, which is UAA. So when that happens, that means, okay, everything downstream from this won't be translated, right? It's not going to be part of the protein. So that means that in this case, right, the only amino acid that we got in this whole translation process was methionine. Because once you get a stop codon, like UAA, nothing from here on out is going to get translated into protein. So you just cut off a huge part, a huge chunk of the protein strand. Okay, so that's the bad thing. So this is actually going to be, we can actually erase all of this, right? Since this isn't glutamate anymore, it's a stop codon, which means we're not going to transcribe or translate any of these amino acids downstream from the stop codon, right? So we just cut off a huge chunk of our protein end product, right? Our protein end product. So that's basically what a nonsense mutation is, which is why it can affect you significantly, right? If you have a nonsense mutation, like imagine if this strand, right? Imagine if we had right here, like imagine if we had the C here, right? But this strand was huge, right? So imagine if we had, like, instead of 3 amino acids like we saw, imagine if we had like 20, and you had a stop codon on the second repeat, right? So we had AUG and then a stop codon, then all of the 19 would be cut off, right? We wouldn't have those 19 amino acids part of your protein. Okay, so that's a nonsense mutation. When you replace a normal codon that codes for amino acid, and you replace that with a stop codon. So what are your stop codons, right? So one we just saw, which is UAA, then another one is UAG, and another one is UGA, okay? These 3 will stop translation, and anything downstream, all those amino acids, won't be incorporated into the protein, okay? So that's a nonsense mutation. And then next we'll talk about a frameshift mutation. All right, so see you guys in the next video.